In the remote wilderness of Chilean Patagonia, a remarkable discovery was made in Aaya cave, where the skeletal remains of an individual were uncovered. Dated to approximately 5,100 years ago, this person belonged to a maritime hunter-gatherer society that inhabited one of the southernmost areas of the ancient world, a place still permanently occupied by humans today. The fossil, designated as A460, was recovered from the Aaya site in the western Patagonian archipelago, a maritime region marked by fjords and glacial channels. Despite the challenges of this cold, wet climate and its geographic isolation, early inhabitants of this area had developed remarkable adaptations for survival. This environment posed immense difficulties for human habitation, yet archaeological and genetic evidence reveals a thriving maritime culture that relied on rich marine resources, such as seals, fish, and mollusks. Early European explorers were amazed by the resilience of these people, who could endure freezing temperatures with minimal clothing and no campfires. Radiocarbon dating places the Aaya woman in a critical time when specialized marine adaptations were flourishing in the region. Her existence sheds light on the deep genetic legacy of early South Americans, offering a glimpse into their remarkable resilience and survival skills. The Aaya woman's genome connects her to the later Khazar and Yam peoples, indicating that these southern populations maintained a distinct cultural and genetic identity for at least 5,000 years. The genome of the Aaya woman is part of the broader southern Native American lineage, which split from East Asians and Siberians around 23,000 years ago, likely during a period of isolation in Beringia. This discovery not only deepens our understanding of early South American societies, but also connects these ancient people to the broader story of human migration across the Americas. A sub-branch separated and migrated southward through the Americas, eventually reaching as far as Tierra del Fuego. The Aaya woman genome, like others from this region, shows high affinity to other ancient Patagonian and Tierra del Fuego populations. Interestingly, these individuals appear equidistant from their terrestrial contemporaries, indicating a split between terrestrial and maritime lifeways sometime after initial settlement, but before 6,000 years ago. While the maritime groups adapted to cold coastlines and high-protein diets, inland groups relied more heavily on terrestrial game and plant resources. Crucially, Aaya's genome, recovered from a tooth, carries signals of archaic admixture, specifically introgression from Neanderthals and Denisovans. These signals are subtle but consistent with known global patterns of archaic ancestry. All non-African populations carry around 2% Neanderthal DNA, and many Asian-derived lineages, including Native Americans, also contain traces of Denisovan DNA. The Denisovan signal is of particular interest in Patagonia, as some ancient individuals, particularly from Uruguay and Panama, have been shown to carry more Denisovan than Neanderthal ancestry. The presence of Denisovan ancestry in South America has profound implications. Originally thought to be confined to Southeast Asia and Oceania, Denisovan DNA has now been confirmed in small amounts among Native Americans, especially those deriving from the southern Native American lineage. This suggests that Denisovan admixture occurred in East Asia before the ancestors of Native Americans crossed into the Americas via Beringia. The fact that Aaya, far to the south in Patagonia, retains even faint echoes of this lineage testifies to the remarkable durability of that ancient encounter. Moreover, the Denisovan signal is not evenly distributed across the Americas. It is found in greater abundance among some ancient and modern South American groups than in northern populations. This pattern may reflect the rapid dispersal and partial isolation of certain founding populations who carry more Denisovan ancestry, especially those who move swiftly along coastal routes. Such a dispersal would be consistent with archaeological and genomic models indicating rapid southward expansion as early as 14,000 years ago. The possibility that Denisovan ancestry contributed to the robust physique seen in Patagonian fossils such as Aaya opens new interpretative vistas. The Denisovans, based on fragmentary fossil remains, appear to have been large-bodied hominins adapted to cold, high-altitude environments such as the Tibetan Plateau. If their genes entered the human gene pool in East Asia, it is not unreasonable to suppose that some of these traits, strong bones, cold resistance, large lung capacity, were passed on and preserved in extreme environments like southern Patagonia. The Aaya skull exhibits unusually thick cranial bones and pronounced muscular attachments consistent with other finds from Tierra del Fuego and southern Patagonia. This skeletal robustness has long puzzled anthropologists, and it featured prominently in early European accounts of the region. In the journals of Magellan's voyage, the inhabitants of this region were described as giants, often claimed to be nearly 6 feet tall or more, an exaggeration, but grounded in real physical distinction compared to the average European height of the time, about 5.5 feet. Many Patagonian individuals are indeed taller and more heavily built. This robustness is not unique to Aaya, but part of a broader regional pattern among the Yamana and Kesa populations. What might account for it? While environmental factors such as high-protein diets, constant exposure to cold, and strenuous physical activity certainly played a role, genetics must also be considered. As discussed above, Denisovan ancestry may contribute to skeletal density and strength, while Neanderthal mixture is associated in some populations with increased bone growth and muscle mass. While the exact gene variants responsible have yet to be identified in these individuals, the hypothesis remains highly compelling. For instance, genes related to bone density or muscle mass, potentially inherited from archaic hominins, could have been advantageous in the cold, windy conditions of Patagonia, contributing to the thick cranial vaults and robust frames observed. Yet no direct evidence currently supports this hypothesis for Patagonian groups. Genetic studies would need to identify specific loci associated with cranial robusticity and determine if they have archaic origins. Given the complexity of polygenic traits like skull morphology, which are influenced by numerous genes and environmental factors, establishing such a connection is challenging. Moreover, the robustness observed in Patagonian skulls is consistent with patterns seen in other cold-adapted modern human populations who do not have elevated levels of archaic ancestry, suggesting convergent evolution rather than archaic introgression. For A460, the study provides additional context by placing it within a broader genetic framework. The researchers note that A460, along with other ancient Sumaduro samples from Brazil dated to around 10,000 years ago, forms a clade that shares deep ancestry with Australasian populations. The Y chromosome haplogroup inherited on the male lineage is QN848, also linking it to the Sumaduro fossils from Brazil. What's more, the Australasian ancestry is only found on the Atlantic coast of South America, except for one sample found in Panama. This is really perplexing, especially because the oldest samples are over 10,000 years old, suggesting that the population with Australasian ancestry is more ancient than other Native American groups. Ponder on that for a moment. However, the study also highlights a lack of shared cultural features among these samples, suggesting that the genetic signal does not necessarily correlate with cultural practices. This is significant for A460, as it indicates that while its ancestors may have carried traces of Australasian ancestry, this did not manifest in distinct cultural traits that can be archaeologically identified, such as specific burial practices or tool technologies. The presence of Australasian ancestry in A460 and other South Americans raises questions about the migration routes and timing of this genetic contribution. Both studies agree that the signal likely entered the Americas via the Pacific coastal route, a pathway that allowed early migrants to move quickly into South America. The study estimates that this migration occurred around 15,000 years ago, with radiocarbon dates from sites like Monte Verde in southern Chile, 14,800 years ago, supporting the rapid dispersal of these early groups. The study further refines this model by proposing that the Australasian signal was introduced before the formation of distinct Amazonian branches, likely in a coastal population that later spread eastward. One intriguing aspect of these findings is the absence of the signal in North and Central American indigenous groups. The study suggests that the migrants carrying the signal may have bypassed these regions, sticking to the Pacific coast and reaching South America without leaving a genetic legacy in the north. Alternatively, the Santos study posits that later population movements, such as those driven by European colonization, may have diluted or erased this signal in northern Central America. For A460 located in Patagonia, this coastal migration model aligns with its geographic position near the Pacific. The Australasian ancestry in A460 also prompts speculation about the physical characteristics of these early Patagonians, particularly their robust skeletal features. Patagonian skulls are noted for their thickness and robustness, with pronounced brow ridges and large mandibles. While these traits are likely adaptations to the cold, windy climate of Patagonia, the presence of Australasian ancestry raises the question of whether genetic contributions from related populations such as indigenous Australians or Melanesians might have influenced these features. Indigenous Australians and Melanesians, who share a common ancestry with the Australasian signal, are known for their robust skeletal structures, a trait often attributed to their adaptation to diverse environments across Sahul, the prehistoric landmass uniting Australia, New Guinea, and Tasmania. However, the genetic contribution of Australasian ancestry to South Americans like A460 is relatively small.